The brain is the most important organ when it comes to human evolution. It suffered two processes. First, encephalization, the temporal and parietal lobes increased and the cerebellum decreased. It was common to all apes and increased the capacity to adapt to the environment. Second, humanization involved many changes that we'll explain later and allowed humans to diverge from apes.
Adaptive mechanisms involve epigenetic mechanisms. Comparative studies have identified transcriptional differences between humans and chimps that include epigenetic modifications in regulatory regions. Demethylation of cytosines in CpG sites is known to be the main epigenetic modification in genomic DNA. It consists of adding a methyl group to a cytosine, a modification that is heritable through cell division. It is established that 12% to 18% of genes that are expressed differently in humans and chimps present methylation differences too.
Some changes in protein sequence may contribute to brain traits specific to humans. One area of research relates to genes associated with microcephaly. These genes appear to function in cell cycle control, and their ability to affect brain size is likely due to their role in regulating the proliferative potential of neural precursor cells during embryogenesis. There are other genes that are involved in brain development, and failure in them can have serious consequences.
The sequencing of complete genomes from different primate species coupled with new technologies has facilitated the identification of gene duplication and gene loss events during human evolution, some of which could have contributed to human brain evolution. An interesting example is the MYH16 gene encoding the myosin heavy chain 16 protein. In primates, it is expressed only in the muscles of the head, including those responsible for mastication, whereas in humans, a frame sheet mutation resulted in the loss of function of this gene, resulting in an undeveloped mastication system. Thus, the loss of this gene might have enabled the relaxation of masticatory muscles, coupled with positive selection for increased brain size.
It is thought that changes in gene expression may have contributed to the development of human brain. Several studies have compared cis-regulatory ratios of brain-related genes across many species, concluding that different cis-regulatory elements of these genes might have experienced positive selection during human evolution. Other studies are focused on comparing patterns of gene expression in the brain across various species. One of its main conclusions is that total gene expression in the brain has been upregulated during human evolution. This agrees with the complexity of our brain, since more developed patterns of expression require a higher regulation.
An emerging area of research is the identification of rapidly evolving non-coding RNAs. A seminal finding in this field was the discovery of HAR1, a long non-coding RNA expressed in neurons. It contained 18 changes in the human lineage since the divergence from chimpanzees. Thus, it is suggested that the human-specific changes in HAR1 might have played a part in the evolution of the human cerebral cortex.
Now we know some of the genetic mechanisms responsible for brain evolution, but is it still evolving? One study identified 2 genes involved in brain function that are positively selected for in humans, and they show that this selection is still going on today. But what does that mean? We still don't have all of the answers, and we don't know how the brain will change in the future, but what we know is that there is still a lot to discover and that we can contribute to it.